Hi everyone. Today we are discussing about prokaryotic replication elongation stage. We have already discussed about the first stage prokaryotic initiation in the previous video. If you have not watched it, we have already pinned the video above, so go and watch the first video before entering to this. During elongation, the DNA polymerase III extends to daughter strands as the replication complex known as replisome travels along the length of the chromosome. So, elongation is done by a multi-protein complex known as replisome. Replisome contains primosome and DNA polymerase III holoenzyme. Primosome contains helicase, primase, and single-strand binding proteins. The DNA polymerase III holoenzyme contains DNA polymerase III core enzyme, sliding clamp, and clamp loader. Now we are coming to DNA polymerase III. This is the major polymerase involved in the replication, and it is a dimer with two similar multi-subunit complexes. It is a DNA-dependent DNA polymerase with high fidelity and accuracy, and also it is a processing enzyme. The major active site is involved in the synthesis of DNA, and its reaction is based on primer extension. So, this is the major reaction occurring during primer extension. DNMP plus DNTP gives DNMPN plus 1 plus pyrophosphate. This is the synthesis of phosphodiester bond reaction. And the second one is this pyrophosphate plus H2O gives 2 inorganic phosphate. That is, this is the hydrolysis of pyrophosphate. So, its energy change delta G is highly negative. And altogether the reaction becomes DNMP plus DNTP plus H2O gives DNMP n plus 1 plus 2 inorganic phosphate. So DNA polymerization is a coupled reaction where the synthesis of phosphodiester bond is coupled to hydrolysis of pyrophosphate. So delta G for phosphodiester bond formation in the first reaction is very close to zero, making it easily reversible. By coupling this reaction to the highly exergonic hydrolysis of pyrophosphate, the second one, the overall free energy change becomes highly negative. So it makes nucleotide incorporation irreversible. All polymerization require nucleotides as triphosphate to act as a substrate. To increase the rate of nucleophilic attack, the nucleophilicity at 3' end must be increased and negative charge on DNTPs should be decreased. So, the active site of the enzyme contains two metal ions, mainly metal ion A and metal ion B. Generally, they contain magnesium. In DNA polymerases, both are prosthetic groups, that is, they are permanently attached. So metal ion A interact with the 3' OH group of primer, decreasing their pKa value. So when pK value decreases, its Ka value increases and 3' OH deprotonates as 3' O-, becoming a strong nucleophile. Meanwhile, metal ion B interact with deoxy-NTP and reduces the negative charge on phosphates. They stabilizes the leaving pyrophosphate and facilitates their hydrolysis. So this is the reaction. Coming to core enzyme of DNA polymerase III, minimum component required for activity of an enzyme is called as core enzyme. DNA polymerase III core enzyme consists of 3 subunits, alpha, epsilon, and beta. Alpha contains active site of enzyme, and the structure of alpha subunit resembles a partially folded right hand. They have 3 domains: finger, thumb, and palm. Palm domain contain active site and they form extensive electrostatic interaction with template DNA. Some domain which are not involved in replication, but they interact with the nascent duplex form of DNA. It helps to maintain contact with the template DNA even when the active site temporarily dissociates after each nucleotide addition. So it helps to continue polymerization from the same site. So it contributes to the processivity of DNA polymerase. In the case of finger domain, they contain a critical alpha helix called O helix that is involved in catalysis. It has basic residues that interact with phosphates of incoming TNTPs and align them parallel. A tyrosine residue that is hydrophobic flip out the next templating base by 90° through hydrophobic interaction. It marks the site for next nucleotide addition and is called as pre-insertion site. In the absence of DNTP binding, O helix is aligned away from bound domain, giving an open conformation for polymerase. So a crevice is visible between thumb and finger domain. Upon DNTP binding, a conformational change creating forward movement in the thumb and a rotational motion in finger closes the crevice. It is called closed conformation. So during the conformational change, the tyrosine amino acid move away from templating base, which return them to their original orientation. If they base pair with the incoming TNTP, they will be in ideal position for coordination with metal ions, which catalyzes the formation of phosphodiester bond. This is the reaction. So, DNA polymerase can distinguish between correct and incorrect DNTP. This is by means of kinetic selectivity. Incorrect DNTPs do not base pair with the template, so they are misaligned in the active site. No coordination will be there with the metal ion B. So catalytic rate abnormally drops and they are eventually outcompeted by correct DNTP that provide greater rates. So this mechanism of selecting substrates based on the velocity they provide are called kinetic selectivity, which is different from a normal enzyme which show geometrical specificity. In the same manner, DNA polymerase can distinguish between DNTPs and RNTPs. So it is by means of bulky side chain amino acids like glutamate in the active site called discriminator amino acids. These amino acids like glutamate can easily accommodate a DNTP without clashes, whereas 2' OH group of RNTP will clash with glutamate, misaligning RNTP. So it slows down the reaction. Accordingly, they can distinguish between DNTPs and RNTPs. DNA polymerase 3 proofreading activity. Mainly, they have proofreading activity in the direction 3' to 5', opposite to polymerization. As we all know, the polymerization will be occur in the direction 5' to 3'. So proofreading activity or exonuclease activity will be in the direction 3' to 5'. And the theta is the regulatory subunit which stimulates proofreading activity. And the proofreading activity resides in epsilon subunit in the DNA polymerase 3. So epsilon subunit has this 3', 5' proofreading or exonuclease activity, and this is a metal mediated catalysis involved in hydrolysis of phosphodiester bond. So incorrect nucleotide incorporations create mismatch creating flaps inside active site, and in the active site the affinity of alpha subunit into such flaps are low. So the flaps move into nearby exonuclease site in epsilon and template DNA remain bound in the active site. So the digestion of flap will be occur and regenerate a proper PT junction there which has higher affinity to active site. So thus polymerase restart DNA synthesis. Sliding clamp in prokaryotes are called as beta clamp, and they are involved in DNA repair, recruitment of histone in eukaryotes, primer removal, DNA condensation, nucleosome synthesis, etc. They are the most contributing factor to the processability of DNA polymerase. It has a ring-shaped structure, which is a homodimer, beta beta dash, and they have a central cavity which can accommodate double-stranded DNA with one or two water molecules. Beta clamp is loaded onto PT junction by clamp loaders. We'll be dealing clamp loaders later. They homodimerize by hydrophobic interaction, and it is a straight conformation because during hydrophobic interaction each half ring extend decreasing their curvature. So water molecules provide a frictionless layer that allow the clamp to slide along DNA freely. After the synthesis of 50 to 60 nucleotides by DNA polymerase, these DNA polymerase physically dislodge from the template. But due to their interaction with the clamp, it is not released into the solution. So DNA polymerase will rebind at the same PT junction from which they dissociate earlier. It facilitates continuous DNA synthesis without gaps. That's why they are the most contributing factor to the processivity of DNA polymerase. Clamp loaders in prokaryotes are called as gamma complex. It is an AAA plus protein, and minimum components that are required for clamp loading is called minimal clamp loader. It exists in two forms mainly, gamma 3 delta delta dash and tau 3 delta delta dash. Gamma 3 delta delta dash is seen free in cytosol and they are involved in clamp unloading. This gamma contain an ATPase activity, mainly SRC motif, and coming to tau 3 delta delta dash, these form seen with holoenzyme, that is they are part of replication and they are involved in clamp loading. Their holoprotein consists of tau 3 delta delta dash psi 5, and both tau and gamma are encoded by the same gene called DNAX. Complete transcription of DNAX gene produce tau subunit, whereas half transcription produce gamma subunit. So gamma lacks the carboxy terminal region of tau subunit. So the carboxy terminal of tau is very critical in determining the flexibility of replication. This tau interact with helicase as well as with DNA polymerase 3. And each tau subunit contains an ATPase active site with an arginine finger as catalytic motif. Delta is a hydrophobic subunit that can interact at any of the dimer interface in the beta clamp. It disrupts hydrophobic interaction between beta and beta dash. Their curvature will increase opening of the ring. Delta dash sequesters the delta in the absence of ATP binding in tau. Chi and psi destabilizes interaction between primase and SSP proteins and also helps in the recruitment of clamp into the newly synthesized PT junction. The steps occurring during the clamp loading are ATP bind each active site and a conformational change exposes delta. Delta binds beta beta dash and their clamp opens. The resulting complex show affinity to a PT junction and a new complex consisting of clamp loader, clamp with bound PT junction forms. This induces a conformational change bringing arginine finger in correct orientation to catalyze ATP hydrolysis. A conformational change sequesters delta by delta dash and beta clamp close around PT junction and clamp loader is released from PT junction. The next DNA polymerase enzyme in prokaryotic DNA replication is DNA polymerase 1. They are involved in primer removal and gap filling due to the primer removal. It is the 5' 3' exonuclease activity that hydrolyzes the last phosphodiester bond between a ribonucleotide in the primer and deoxyribonucleotide in the DNA. These events are mediated by the interaction of polymerase 1 with beta-clamp. Other two enzymes involved in DNA replication are RNase H and ligase. RNase H is involved in primer removal. They hydrolyze phosphodiester bond between ribonucleotide in a RNA-DNA hybrid, mainly PT junction. Ligase, which is involved in nick sealing, it uses energy for their functioning. Mainly they are of two types, NAD-dependent ligase in prokaryotes and ATP-dependent ligase in eukaryotes. The steps involved in ligase function are: first, adenylation of ligase. A nucleophilic attack from amino group of lysine in ligase onto phosphoanhydrate bond in NAD or alpha phosphate group in ATP. An adenylated enzyme intermediate forms here, and energy conserved in the phosphoanhydrate bond is transformed to energy in the adenylated enzyme. Second one is adenylation of DNA. Transfer of AMP from enzyme to 5' phosphate in the nick. And third one is the formation of phosphodiester bond. So here in the nick, but it's formed by the nucleophilic attack from 3' OH group in the nick on to the AMP 5' end. Thus, AMP is released. Replication fork. A Y-shaped junction where active DNA synthesis occurs is called replication fork. A replication fork always moves on duplex region, mainly 5' to 3' direction. The prokaryotic replication genome is called theta model replication because the replication intermediate resemble a theta. Also, replication prokaryote is bidirectional, which means two replication forks will be generated from an origin which moves in the opposite direction. Strand synthesis at replication fork involves synthesis of a DNA in the direction of replication fork movement is called leading strand, whereas another DNA synthesis in the opposite direction at replication fork is called lagging strand. To achieve synchronous replication of both strands, replication proceeds in a semi-discontinuous way. Here, leading strand synthesis is continuous and lagging strand synthesis is discontinuous. Replisome components include primosome and DNA polymerase III holoenzyme. Primosome consists of DNAB helicase, DNAG primase, and SSB single-strand maintenance proteins. And DNA polymerase III holoenzyme consists of DNA polymerase III core enzyme with 3 subunit alpha, epsilon, theta, and clamp loader gamma complex and sliding clamp beta clamp. The major events at replication elongation are explained by trombone model. During initiation, one of the core polymerase in the holoenzyme is recruited to the PT junction. Only a single primer is enough to synthesize leading strand, whereas lagging strand requires many. As primers temporarily interact with helicase, they synthesize primer in the opposite direction to replication fork movement. The PT junction is recognized by gamma complex which lock beta clamp. Beta clamp recruit the second core polymerase in holoenzyme and they synthesize the opposite direction of replication fork. It creates a double-stranded loop called trombone loop. The newly synthesized discontinuous fragments are called Okazaki fragment. Size of the Okazaki fragment depend on affinity between primase and helicase. If the primase helicase affinity is higher, there is frequent primer induction, and this is seen in eukaryotes, so the Okazaki fragment size will be lower, around 100 to 400 nucleotides long. And when primase helicase affinity is lower, their Okazaki fragment size is large, as they are seen in prokaryotes. Around 1000 to 2000 nucleotides can be seen. Okazaki fragment maturation. DNA polymerase III alpha subunit contain an OP domain that interact with template DNA and facilitate their binding. When synthesis of an Okazaki fragment reaches the 5' end of Okazaki fragment that is already synthesized, the OB domain is unable to bind DNA as there is no single-stranded DNA upfront. Their affinity to template decreases and these DNA polymerase III will dissociate, leaving the clamp. Next, RNase H associates with the clamp and removes the primer, and later DNA polymerase I will associate the clamp and they hydrolyze the last ribonucleotide and deoxyribonucleotide phosphodiester bond and fills their gap. Finally, ligase interact with clamp and joining the fragments. So, these are the steps in the Okazaki fragment maturation and the last steps of elongation. Due to the discontinuous nature of lagging strand synthesis, spontaneous mutation rate in lagging strand is lower compared to leading strand. So fidelity is more for lagging strand than leading strand. So that's all about elongation. We will be dealing with termination and regulation of prokaryotic replication in the next video. Stay tuned for that. If you like this video, please like, share, comment, and subscribe our YouTube channel, Ligase Academy, your educational catalyst. Thank you.